So as we discussed in the last video, our cells have these sort of protein frames called major histocompatibility complexes, or MHCs, that can hold examples of proteins in them, like a frame. They hold it out for inspection by other cells. And there are two main types. And class 1 MHCs, all of your cells that are nucleated have them. And they just allow you to display a set of proteins that you normally have inside your cells that you're producing based on your DNA, and you display them to your immune cells, like your cytotoxic T cells or natural killer cells, and say, hey, look, you can't lie. These are the proteins I'm making. That means I am one of your cells, right? But if you've been virally infected, you're going to have viral DNA and you're going to make slightly different proteins, and so that's going to show in your MHC1 here. Or if you are cancerous, right, that's going to show here. And so all of your cells have MHC1s, and those you display to cytotoxic T cells. MHC2s are only found in phagocytes, phagocytic cells, specifically macrophages, which tend to devour things like bacteria or viruses, digest them up, and then take bits of these proteins and display them like a trophy. Look what I have killed, right, in the plasma membrane of their cell. And they display these not to cytotoxic T cells, but to helper T cells. And they say, helper T cells, right? You're the generals. You run the immune response show. This is what I've found. This is infecting us. Is this the one you're looking for? Because all helper T cells are looking for a very specific antigen. And if it's not, the helper T cells will move on. But if it is, the helper T cells will ding, 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 set off the alarms, and they will multiply and they will activate other cells. And this is what the process of antigen presentation looks like, or antigen recognition, which is sort of that same process from the perspective of the T cell. So you have your MHC, either 1 or 2, depending on what situation this is. And you have to have an antigen recognition protein, which allows you to bind to the particular MHC. And then there's an antigen receptor on the T cell that you're going to put up with the antigen to see if they fit. Right, see if they're the appropriate shape. And if they are, the inactive T cell will become an active T cell and will respond, right? So we've talked about these MHCs, but again, you also need an antigen recognition protein from the T cell. And there are two classes. CD8 markers are the markers that will help you bind to an MHC1. And so these are found in CD8 T cells, or cytotoxic T cells, which will bind to MHC1s and check out, what kinds of proteins are you making? Are they yours, or are you virally infected or cancerous, et cetera, et cetera? CD4 markers are found in helper T cells, I should say, and they bind with MHC2, class 2 major histocompatibility complex. And so CD4 markers found on helper T cells bind with the MHC2s on macrophages, antigen-presenting cells, and help you see whether what they've eaten is what you're looking for as a helper T cell. So here's a look at an inactive CD8 cell, again, very often a cytotoxic T cell, binding with an infected cell. This infected cell, in its MHC1, is displaying an antigen that probably includes some proteins from viral DNA. This T cell is going to bind to it with its CD8 marker, and this is going to activate it and cause it to start dividing and dividing, so there are more of it to attack these kinds of cells. So in addition to cytotoxic T cells, you're also going to make memory T cells, which do not differentiate further after that first exposure. They wait until the second exposure, and they're long-lived. And this means essentially they'll stay in your body for a long time in case you ever get infected again, and they can protect you then. And then also suppressor T cells, which can secrete suppression factors eventually to slow down the T and B cells at the end once you've sort of defeated the infection. Now, helper T cells, which are CD4 T cells, right, they'll bind to an antigen-presenting cell that has eaten something, a bacterium or a virus. And they have this CD4 marker that will help them attach and will cause co-stimulation. And if they're binding to the correct antigen, that will activate this helper T cell and cause it to multiply. And it will also produce some active helper T cells, which will then immediately begin, in addition to multiplying, secreting chemicals, right? That will communicate with the rest of your immune system and organize it and cause a big immune response. They will also make memory T cells, which are inactive until the next time that they get exposed to this same antigen. And so again, they're long-lived. They'll stay in your body for a long time and make you essentially immune to this particular disease that carries this antigen for at least a while. Now, B cells also have to be sort of sensitized and then activated by being exposed to the antigen and then subsequently to a T cell. So essentially, they will bind to this antigen, right? These things that look like antibodies, because they essentially are antibodies, they just are sticking off the outside of the cell. These are really their antigen receptors, right? And when they bind to the antigen receptors, that means they're the appropriate shape. They're the antigens these B cells are looking for. We have lots of different kinds of B cells, remember. They're very specific in what they'll bind to. But if this is what they're supposed to bind to, they'll bind to it. They'll, through endocytosis, absorb these antigens and then place them on the outside in their MHC2. So just like macrophages, they have their own MHC2. Now, at this point, they've been sensitized, but they haven't been fully activated yet. And they'll be activated when they come to a helper T cell and they display the appropriate antigen to the helper T cell. And the helper T cell will then secrete chemicals that will activate the B cell and cause the B cell to begin multiplying itself and making antibodies, which is what B cells do. So in the same way that when T cells are activated, they begin multiplying and multiplying and multiplying to start a huge immune response, B cells will do the same. They will multiply and multiply and multiply, and they'll make two main kinds of cells. They'll make active B cells, which become something called a plasma cell, and those make antibodies. And you can see antibodies are proteins, and these plasma B cells are full of rough endoplasmic reticulum, which is full of ribosomes that make proteins, right? All these are here for, really, is to make these antibodies. And so when you look at them under a microscope, you just see tons of rough ER. But the other type of B cell that becomes activated, that those multiplying B cells will become, is a memory B cell. And just like memory T cells, both cytotoxic and helper memory T cells, these are inactive until the next time that they're exposed to this particular antigen. They are long-lived, and they mean that the next time you get infected with that particular pathogen that has that particular antigen on the outside, they will have a very fast recognition and fast production of antibodies. So these antibodies that are made by these plasma B cells, what do they look like? Well, there are different kinds, but all of them have these things in common. They have a pair of heavy chains and a pair of light chains. And we'll look at a picture in a moment. And each pair has a constant segment that is the base of the antibody and is always the same. But they have on the end a variable segment. And this is different in every antibody because it's the antigen binding site. And different, variable segments will bind with different antigens. And what does the variable segment look like? Well, it depends upon the antigen receptors on the B cell that made it. They look just like the antigen receptors on the B cell. So if a particular antigen activated a B cell, the antibodies that B cell makes will bind to that same antigen, which makes sense, right? You wouldn't want to activate a B cell for H1N1 and then have it attack, H2N6. So here's a look at one kind of antibody, and this is the heavy chain here in the middle, right? This purple thing. This is the light chain out here. Literally just the heavy chain is larger, the light chain is smaller. And down here, this part of both chains is the same, right? Doesn't matter what antibody you're looking at, it's always the same. But out here, this is the variable segment, which contains the antigen binding sites. And this is what's different between different antibodies, again, depending on which B cell made it and what their antigen receptor looked like. Now, every antigen has on it these antigenic determinant sites. They're sometimes called epitopes. And they are little sites that the antibodies will bind to. And again, mark them for the destruction. But you may recognize this from when we talked about blood antigens and antibodies. These antibodies have two receptor sites. So sometimes one receptor site will bind to an antigenic determinant site on this antigen, and the other one will bind to another antigen, right? So, and you may end up getting like two bacteria or two viruses stuck together then. Just like the way that your red blood cells will clump up, agglutinate really is the term, when they're attacked by antibodies. So often will pathogens that have particular antigens on them. And there are a lot of different kinds of antibodies. Some of them attach to basophil and mast cell surfaces, immunoglobulin E, and cause inflammation. Some bind on B cell surfaces and help sensitize B cells. Some are involved in allergies. Most of them are immunoglobulin G. So antibodies are sometimes called immunoglobulins because they're globular proteins for immune purposes. Immunoglobulin G, 4/5 of all antibodies, and these are what are responsible for resistance against viruses, bacteria, toxins. This is mostly what we're talking about when we've been talking about these antibodies. And you can see they've all got slightly different shapes, but they all have a heavy chain and a light chain, and they all have a variable segment at the end that will bind to different antigens. Now, as you might expect, that primary immune response, the first time you get infected with something, it takes a while to really build up your response. Because first of all, you need your T cells and your B cells have to come into contact with the pathogen and with the antigen that's on the outside of its cells. And that doesn't happen immediately, right? Because you have very few cells that are looking for any particular antigen. Now, it helps that they focus in your lymph nodes, for instance, in your spleen, places where we know that we're routing fluid that probably has whatever's infecting you. But it takes a while for those macrophages to eat it up, find the right T cell, do the antigen presentation, and the T cell's got to activate the B cell that's been exposed, and then the B cell has to start multiplying and eventually make a lot of antibodies. So this takes a week or two, and this is why when you get sick with a cold or flu or something, it usually takes a week, 10 days before you really get better, sometimes longer. Now, your secondary immune response. This is when you've already been infected with something and you get infected with it again, but this time you already have memory B cells and memory T cells. And so you have lots of them, so you're going to detect the virus or bacteria more quickly, and you're going to have a more immediate response by B cells for that particular antigen. And it's an overwhelming response. And sometimes these last decades, right? So sometimes immunity can last a very long time. But this is the main reason why your secondary response is different from your first, right? So these are two different kinds of immunoglobulins, but you can see that they don't start ramping up until mid to the end of the first week that you've been infected, and then they kind of peak in the middle of the second week that you're infected. By then you should start feeling better. But your secondary immune response, the first day you're infected, you're probably already making antibodies, and by the middle of that week, you're already making as many as you ever made here, and you're going to end up making tons, right? The scale is just much larger, and so you'll probably never even notice that you were infected the second time. And so the existence of these memory cells, as well as antibodies, are what you look for if you want to know whether people have developed immunity to a certain disease. And that's why, in addition to stimulating antibody production, a lot of these vaccines, in the tests they're running, they're looking for the stimulation of memory B cells and memory T cells as well, in terms of a vaccine for COVID. So this is the secondary response that you want to see if your vaccine worked.